It all begins with a single cell, a zygote. But how does this tiny, almost invisible cell multiply to form a complete human being? Let's uncover the hidden science of life's very first divisions. Every human life starts from one microscopic cell, called a zygote. This single cell holds all the genetic instructions, the blueprint for an entire body. Inside, it lies the potential to form every organ, every bone, every neuron that will one day think and feel. The journey from one cell to billions is one of the most fascinating processes in biology, and it starts just hours after fertilization. So, how does it all begin? When a sperm successfully meets and fuses with an egg in the fallopian tube, their genetic material combines, creating a single diploid cell known as the zygote. This new cell contains 46 chromosomes, half from the mother, half from the father, forming a complete set of human DNA. According to Langman's medical embryology, this moment marks the scientific beginning of a new human life. Within just 24 to 30 hours after fertilization, the zygote begins its very first mitotic division. This isn't just a simple split, it's a meticulously choreographed dance of life. The single cell divides into two identical cells called blastomeres. Each of these new cells carries the exact same genetic code. Soon after, these blastomeres divide again and again and again. 2 become 4, 4 become 8, and then 16. This rapid multiplication is called cleavage. What's truly interesting is that the embryo doesn't actually grow in size during this stage. Imagine cutting a cake into more and more slices without changing the size of the cake itself. The cells become smaller and more numerous, preparing for the next big step. Around day 3, the embryo starts to look like a tiny cluster of grapes, a solid ball of 16 to 32 cells. This structure is called the morula. These cells are packed tightly together and begin communicating with each other, sending signals that will determine their future roles. Research from The Developing Human by Moore and Persaud shows that the morula enters the uterus around this time, carried gently along by tiny, hair-like cilia inside the fallopian tube. It's a slow, delicate journey toward its final destination. By the 5th day, a remarkable transformation occurs. The morula evolves into a blastocyst, a hollow sphere with two distinct and crucial parts. The outer layer is called the trophoblast, which will eventually form the placenta, the life support system for the growing baby. The inner cell mass is the embryoblast, which becomes the actual baby. A fluid-filled cavity, the blastocoel, also forms inside. This structural organization is a huge milestone. It marks the first sign of cellular differentiation, the moment when cells begin to take on specialized roles, a process that defines the entire course of development. Then, by day 6 or 7, the blastocyst reaches the uterine wall and begins one of the most critical events in early life, implantation. The trophoblast cells on the outside release enzymes that allow the embryo to attach and then embed itself into the soft, nutrient-rich lining of the uterus, the endometrium. This is where it will make its home for the next 9 months. As the American College of Obstetricians and Gynecologists notes, successful implantation is the foundation of every healthy pregnancy, ensuring the embryo receives the vital nutrients and oxygen it needs from the mother's blood. So, what is the science behind all this division? Each time the zygote or its subsequent cells divide, it follows a precise pattern called mitosis. First, the DNA inside the cell meticulously duplicates itself, ensuring each new cell gets a perfect copy. Then, the nucleus splits, and finally, the cell's body, the cytoplasm, divides equally. Because each new cell contains identical DNA, every single part of your body, from your brain cells to your skin, can trace its lineage right back to that one original zygote. Even more amazing, these early embryonic cells are totipotent, which means each one has the extraordinary potential to form an entire organism all by itself. Why does this early division matter so much? because these initial divisions set the stage for all future growth and development. The precision of this stage is everything. Errors during cleavage can unfortunately lead to genetic abnormalities or failed implantation. Recent studies in Nature Cell Biology have revealed that these very first embryonic divisions rely heavily on maternal RNA and proteins, materials that were stored in the egg long before fertilization even occurred. The mother's body provides the initial toolkit. The incredible precision of this stage determines whether a new life will continue developing normally. By the end of the first week, what began as a single cell has become an embryo with hundreds of cells, each one already knowing what it is destined to become. Some are programmed to form the heart, others the brain, and still others the skin. This organized chaos, driven by the intricate language of DNA, chemistry, and time, is truly one of nature's greatest miracles. From one invisible cell to the beginning of a heartbeat, this journey shows the extraordinary intelligence coded within every strand of DNA. The zygote's first divisions aren't just biological processes, they breathe the very spark of life itself. If you found this journey into the beginning of life as fascinating as we do, please subscribe to MedLifeLink for more medically accurate stories about life, biology, and the science of creation. Thank you for watching.